My name is Tim Miller, and I'm hoping to give you a little bit of a different taste of a presentation today. And this was actually a great talk to follow on the last one, so thank you for the last speaker setting me up really, really well about the need for manufacturing and just how we think about AAV manufacturing in this space. So cool thing about technology these days, especially since so many of us are used to doing Zoom meetings or team meetings, we have a little bit of a video just to start. But it's going to give you a little bit of a different flavor and tell you a little bit part of our story. So we'll roll to the next slide. All right. Can you guys roll that? It began with a question. What if? What if we could find a way to accelerate access to rare gene therapies for the patients that need them the most? What if we could find a way to reduce or break through the bottleneck in gene therapy manufacturing? But we didn't stop there. We got started.
Since Forge Biologics launched in July of 2020, we've closed 2 investment rounds totaling $160 million. We've hired over 100 people in the first year, growing our world-class team to help set a new standard for client experience within the AAV gene therapy manufacturing space. We are busy serving clients and preparing additional dedicated GMP manufacturing capacity to meet the demands of this explosive industry. And every day, we get one step closer to achieving our mission of enabling access to life-changing gene therapies and bringing them from idea into reality.
What we're building here at the Hearth, our AAV manufacturing center, will long outlive any of us that are working here. For decades, the promise of gene therapy has provided hope to families around the world. And we wanted to help forge alliances with all the stakeholders, from families to clients to other manufacturers, to clinical sites and hospitals around the world, to be able to provide access to novel, potentially life-saving gene therapies. What we're doing is manufacturing gene therapies for life. So we have a little bit of a flavor at Forge about we're all trying to be superheroes. And while we work with many of our patients, part of what our job is they're already heroes. We're just also trying to help make them super. And so when we think about the AAV manufacturing that needs to go on and for the rest of the space, really was the origin story for the Forge model. And so we're a hybrid gene therapy company. We're a CDMO primarily, but also have a small therapeutics pipeline. And part of the fun thing about assembling a team of superheroes is really bringing in leadership from the gene and cell therapy field. And having been in gene therapy for a little over 20 years, the success stories have all been because you've brought people in that know what they're doing, that have experienced both the therapeutics and manufacturing arms. And so to highlight a couple of our team members, many of whom are here today, David Dismuke was at UNC and ran a lot of the manufacturing core there, and then went on from Voyager and Stride to come to Forge. Jason Eichholz ran all of the manufacturing at Nationwide Children's Hospital for a number of years. John Maslowski was CEO of FiberCell in Castle Creek for a while. So John built out a lot of his own facility. Maria Escolar is our Chief Medical Officer and is a world-renowned rare disease expert. You guys all missed the cool video. You just missed the cool video. It's probably available on our website. And just, really one of the keys to success is having over 150 years worth of leadership in the gene and cell therapy field. Many of us have built out our own pipelines and have built out manufacturing centers. And so In July of 2020, we were 4 people, and we closed a Series A. We raised 120 million Series B less than a year later, and now we're up over 120 employees, many of whom have worked with us previously in different spaces. So we kind of covered a little bit of that, but part of the key things and the tools that are important for us to develop and serve both our clients and our patients is really how we think about the cell lines. And it was a great talk right before this that highlighted a lot of the triple transfection methods and talked about the cell lines. Having developed a variety of different gene therapies, one of the key things when you're working with a lot of manufacturers is how do you think about your 293 cell line? And so we did this from the beginning. We were able to secure the cell line, our licenses, and we adapted it to serum-free media, grew it up to suspension cultures, and have the ability to now grow it into large scales. We grew the team. We've advanced a small clinical pipeline. We're now, our lead product is in the clinic. And we have now, some of the things that helps differentiate us is moving from a research grade all the way through. You can take our cell line, you can come in and do some very quick 1 liter or 2 liter runs if you want, and then you can go in and you can bring that through your engineering, toxicology, and into your clinical, and eventually into your commercial runs too. So a key thing for many of you, and you don't have to raise your hand, but having had to do bridging studies in previous companies, moving your process from an adherent, for example, into a suspension, it's a lot of work. And as we look out into the space, I don't know about all of you, but for the past 2 or 3 years, it seems there has been a lot of money that's been dumped into, really manufacturing, whether it's companies trying to build out their own manufacturing pipeline or their own manufacturing capabilities. But even with a lot of that, if we were to approve, if the FDA were to approve hemophilia and Duchenne, for example, AV programs, it would soak up probably half of the bioreactors on the East Coast right now, just to be able to meet that commercial demand. And I think you saw that earlier as we move into this next generation of gene therapies. And thinking about prime editing, base editing, CRISPR, and how you deliver these technologies, that the need for manufacturing in particular is only going to increase. Part of that's because of the number of clinical trials, right? So even the FDA has come out and said they anticipate approving up to 25 BLA submissions, in the next few years. And as we look out in the 5 to 8 year time frame, and more successful programs go from early stage to late stage to clinical, really you need to work with a great partner as a CDMO. So talking about a little bit of our ignition cells, that's what we call them. It's the HEK 293 suspension cell line. Again, you can come in and use it. It's pretty robust, and we have some surprises, I think, that'll come out in the next couple of months around how we're thinking about using that. Our Ember Plasmid is one that we created. I'll show you just a little bit of data. Some of the presentations here go a little data heavy. We tried to stay pretty light so that people could absorb it. But again, our helper plasmid is one that we made because of some of the concerns in the space. David actually helped make one of the early ones, and now we went back and recreated that and are able to help both our clients and with a plasmid that's both smaller, safer because it doesn't package any adenoviral strands, and is able to produce a little bit of the same thing. So here's just a comparison between 2 of the ones that are out there that are used. And as we think about trying to compare to the one that's most commonly used, the PX80 plasmid, you can see here that, again, our vector purity, whether you're making with X80 or the Ember plasmid, again, you see it by vector transgene as well, that again, things are highly pure and very productive. And again, you can see that when you do a typical transduction, using AAV with, again, a pretty typical 293, it transduces very well. And this really is able to help a lot of our clients accelerate their production using our Blaze system. So if you want, you can go online, you can go in and say, I need to do lead candidate selection. And so you can come in, do some quick AAV using our cell lines, and go ahead and be able to produce for your early stage. One of the key things that many people that we work with need right now is process development and analytical development. And actually, those are where we've hired the most people. It's really the PD and AD teams. By the time you get to GMP, it theoretically should be a lot of shake and bake, right? So what we've done is brought on these teams to help clients so that they can work on their process. We've made AAV 2, 5, 3, 6, 7, 8, 9, 10, 74, MH78. So lots of capsids that have been made, but many groups need, because of their gene of interest, a little bit of work on the PD and AD side. And then as we move into GMP, our systems are fully disposable, single use, and again, we're up to 500 to 1,000 liter scale. So transition just a little bit to tell you a part of our story about an innovative gene therapy pipeline. When we think about bringing pipeline candidates forward for us, it has to be a little bit different. We are starting to look and the field is moving a bit away from single route of administration, single capsid now into multiple routes of administration and even some multiple capsids. So for us, we'll talk a little bit about our FBX 101 program today, just because it's kind of neat, and we'll leave the 201 program for another time. So this is for the treatment of patients with Krabbe disease. It's first of its kind in the world. It's a combination program where we give a bone marrow transplant plus a systemically delivered AAV. Krabbe disease, and I think you might have heard earlier today, is a rare fatal disorder. It's very similar to spinal muscular atrophy, where the patients or the kids progress, and unfortunately it's mortal between the ages of 2 and 3. So it's a systemic AAV administration. Sorry about the text. I'm not sure how that didn't convert very well. But it's a bone marrow transplant, again with a systemic AAV10. Very quick data slides going on. So on the left-hand slide, on the far left, you see, again, the untreated mice die very quickly. The single routes of administration are the orange and, I think, red lines, and then the top line is blue, where you see a combination treatment does better than either route alone. So, and then the one on the right pretty much shows what we've all spent the last 7 or 8 years developing a knowledge base around, which is treat early, treat with more, and generally going to be better for patients. And we do have a couple of dog videos. So there is a naturally occurring dog model. On the left, this is an untreated animal. Let's see if I push it, it plays. So these animals, unfortunately, have a lot of motor dysplasias, neurocognitive effects. You can't really see that in these, but as we went and did, again, an AAV treatment, the AAV treatment alone actually worked partially. It did increase their survival about 3-fold, but the animals still had a lot of neuromuscular impairments. And then so again, these animals reach a humane endpoint. And so when we gave the combination treatment, what we were able to see was these animals did great. If you can see it, whoop. Go back one more time. Well, I guess you'll just have to look on our website. But what was really interesting about this particular treatment was how this changes the way that we think about what's an efficacious and safe dose. Because we give a bone marrow transplant, we actually immune ablate the animals or the patients, so we remove the immune system. Then a few weeks later, we give a systemic AAV when they're still basically under immunosuppression. So we remove a lot of the concerns by some of the immune challenges, but what was really interesting about this data was because there's no immune system, you extended the infectivity period. And so for many of us that have done IND-enabling biodistribution type of assessments, you see usually a peak around day 30 and it starts to come down. We saw almost a 10x jump all the way out to 6 months in all of the organs that we tested. So it changes the way that we think about if you remove some of the immune challenge and you're able to increase the infectivity period, you're actually able to provide functionally a higher dose. So we think that this is a great platform for us to jump off of. It's, again, a combination bone marrow transplant plus a systemic AAV. So I only have about a minute left, and we're happy to take questions in the back, but we do have a cocktail reception where we are going to be celebrating some of the superpowers of gene therapy right out back on the veranda. Thank you.